ultraviolet radiation and ultraviolet absorption spectra of proteins, amino acids, and nucleic acids.
What is ultraviolet radiation? What is ultraviolet radiation? Ultraviolet radiation is the wavelength of light that lie immediately below the end of visible spectrum and cannot be detected by the human eye. It is strongly absorbed by a group of structures as organic compounds that possess aromatic rings as those of the side chains of phenylalanine, one with amino acids, also amino acid tyrosine and tryptophan, or structures that have multiple or conjugated double bonds as those conjugated double bonds present in purine and pyrimidine bases of nucleic acids, the polyunsaturated fatty acids, heme groups, also numerous cofactors including flavins and cyanocobalamin, which have aromatic rings or include a metal in its structure, respectively. Of course, ultraviolet rays have beneficial as well as deleterious effects, as you all know. Of the beneficial effects of ultraviolet rays, it is a participant in the process of vitamin D synthesis, which occurs under the skin, and the extent of vitamin D synthesis by conversion of cholesterol to vitamin D is directly related to the intensity of exposure and inversely to the extent of pigmentation in the skin. And this means that the darker the skin color, the lesser the amount of vitamin D synthesized. But wherever there is sunlight, vitamin D can be formed by the utilization of ultraviolet rays. In temperate climates, as those areas with moderate temperatures, the plasma concentration of vitamin D is highest at the end of summer and lowest at the end of winter. And this is also the case in the latitudes about 40 degrees north or south, as there is very little ultraviolet radiation of the appropriate wavelength in winter. So, care should be taken when using sunblock creams and other skincare preparations, as their inappropriate use may be harmful. They may decrease the sense of vitamin D or may have other deleterious or unwanted effects on the skin. We have the 7-dehydrocholesterol as an intermediate in the sense of cholesterol in the skin, which undergoes a non-enzymatic reaction on exposure to ultraviolet light. And a similar process occurs in the ergosterol in plants and yeast, yielding the previtamin D. When irradiated with ultraviolet light for the 7-dehydrocholesterol in the skin or the ergosterol in plants, the ring B in cholesterol structure is opened to form vitamin D3 in mammals and D2 in plants. This is regarding the beneficial effects of vitamin D and their importance for humans and plants. But the ultraviolet rays has some laboratory and clinical chemistry applications, as we can use an absorption spectrum of ultraviolet rays as well as a spectrum of other colors to determine the concentration of specific analytes or substances. And as mentioned before, compounds having aromatic rings as the amino acid phenylalanine and tryptophan can absorb the ultraviolet rays, and this absorption can be used for the measurement of their presence and their concentration. Different compounds also absorb light at different wavelengths, and this absorbance is proportional to the concentration of the colored compound or the end product during measurements. So we have a question here: why the absorbance of protein solutions is measured as the wavelength 280 nanometers, which is the visible spectrum, although most amino acids do not absorb the visible light. And as you know, that amino acids do not absorb visible light, and so they are colorless. But the amino acid tryptophan, phenylalanine, and tyrosine, which possess aromatic rings, absorb the ultraviolet light as a wavelength of 250 to 290 nanometers, and tryptophan is 10 times more than the latter ones. So tryptophan is a major contributor to the absorption spectrum of proteins at 280 nanometers. Another similar point and a similar question: why the absorbance of nucleic acid solution is measured at the wavelength of 260 nanometers, which is also the visible spectrum. And as you know, nucleotides and nucleic acids absorb ultraviolet light due to their content of the conjugated double bonds in their purine and pyrimidine bases, and absorption is maximum at the pH 7 for the wavelength close to 260 nanometers. So, concentration of nucleotides and nucleic acids thus often is expressed in terms of absorbance at 260 nanometers. In laboratories, we have other techniques that utilize ultraviolet radiation, as we have the RIP technique, which is an RNA immunoprecipitation method, like that of SHIP or chromatin immunoprecipitation, which is used to score specific binding of proteins to a specific RNA in vivo. Also, another technique is CLIP or cross-linking protein-RNA and immunoprecipitation. In this method, also, ultraviolet cross-linking is utilized to induce covalent attachment of distinct proteins to specific RNAs in vivo, so that the RNAs that are protein bound can be purified from cell lysis, and immunoprecipitation is done with subsequent sequencing. But what about the deleterious effects or the damaging effects that ultraviolet can induce? So ultraviolet, which may be used in some laboratory techniques as in protein cross-linking, this short high energy wavelength, the ultraviolet rays, has deleterious effects, as it has a mutagenic effect on DNA due to their absorption by nucleotides in DNA and RNA, as mentioned before. This absorption may lead to cross-linking or formation of thymine-thymine dimers and other chemical modifications. So, also ultraviolet rays can rupture the covalent bonds in proteins and induce cross-linking, as well as the generation of free radicals. All of these are deleterious effects and dangerous chemical change in these compounds. A famous example of the deleterious effect of ultraviolet rays is the induction of skin cancer. But how do they induce skin cancer? And in spite of the weak penetration deep beyond the epidermis. So the ultraviolet radiation does not penetrate deep beyond the epidermis, but the defect occurs. So the defects are often cumulative due to absorption of the ultraviolet rays by DNA and RNA, which, as mentioned earlier, may cause formation of thymine-thymine dimer. And with prolonged exposure will lead to accumulation of the defects or mutations in the DNA. This will lead to accumulation of damage by the skin. And when there is prolonged exposure to sunlight or during times of high ultraviolet concentrations, which may exceed the cellular repair capacity, will lead to development of skin cancer and myelomas. Fortunately, these mutations are passed on to successive generations of cells, but only within an organism. And this means they are not transmitted to generations. Also, one of the deleterious effects or harmful effects of ultraviolet rays in laboratories is that which occurs when there is transient exposure for ultraviolet rays during laboratory techniques for DNA and RNA visualization and gel electrophoresis, which may cause conjunctivitis. And this requires the protection by wearing specific shields for the ultraviolet rays. Thank you.
You can find other relevant information about this lecture and other lectures at this site.